Hello and welcome, dear students. Today we are going to discuss the topic origins of genetic diversity. The main objectives of this module are to know about the concept of genetic diversity, to know about the origins of genetic diversity, to understand importance of genetic diversity, and finally to know about the molecular tools that assess the genetic diversity. Dear students, let's start with the introduction first. Genetic diversity is the total number of genetic characteristics in the genetic makeup of a species. It ranges widely from the number of species to differences within species and can be attributed to the span of survival for a particular species. It is distinguished from genetic variability, which describes the tendency of genetic characteristics to vary. It is a study undertaken to classify an individual or a population compared to other individuals or populations. It quantifies the magnitude of genetic variability within a population, which is a fundamental source of biodiversity. Genes are the fundamental units of biodiversity, the raw material for evolution, and the source of the enormous variety of plants, animals, communities, and ecosystems that we seek to protect, admire, and use. Genetic variation shapes and defines individuals, populations, subspecies, species, and ultimately the kingdoms of life on Earth. Genetic diversity among individuals reflects the presence of different alleles or alternative forms of a gene in the gene pool, and hence different genotypes within populations. Genetic diversity should be distinguished from genetic variability, which describes the tendency of genetic traits to vary within populations. Since the beginning of 20th century, the study of genetic diversity has been the major focus of core evolutionary and conservation biology. The theoretical matrix developed, such as genetic variance and heritability, provided the quantitative standards necessary for the evolutionary synthesis. Further, research has focused on the origin of genetic diversity, its maintenance, and its role in evolution. Simple questions such as who breeds with whom initiated studies on the relatedness of populations. These investigations led to the formation of metapopulation theory, where a group of specially separated populations of the same species interact at some level and form a coherent larger group. The discovery of special structure in populations was a key element in the early concepts and models of population ecology, genetics, and adaptive evolution. How different levels of genetic variation affect the rate of evolutionary change within populations has also been intensively studied. Subsequently, the detection of genetic variation has become more sensitive, firstly through the utilization of variations in enzymes, that's alloenzymes, and then through PCR-based marker systems, allowing direct examination of DNA sequence variations. The precise detection of genetic variation diversity has greatly enhanced the studies of evolution. There is no doubt that the genetic variation influences the fitness of individuals, and that this is reflected in natural selection. In this regard, individual genotypes must vary in ecologically important ways. Ecological adaptation is a significant factor, for example, in range expansion of different species. Species with different genotypes conferring the highest levels of fitness are expected to survive and reproduce better, shifting the gene pool over time towards higher frequencies of the alleles, making up the more successful genotypes reported than when an increase in fitness is allowed. Genetic diversity can increase the population growth rate, but only if the population is not regulated by other factors and if it is experiencing directional selection. Despite the presence of genetic variation in ecologically important traits, relatively little is known about the range of potential ecological effects of genetic diversity on population dynamics, species interactions, and ecosystem processes. This has led to the rise of the field of molecular ecology, which integrates the application of molecular population genetics, phylogenetics, and genomics to answer ecological questions. Information about genetic diversity is necessary for the development of appropriate strategies in conservation biology as well as in many other applied fields. From a basic evolutionary standpoint, genetic diversity is assumed to be crucial for the evolutionary potential of a species. Research programs that aim to investigate population structure provide evolutionary insights into the demographic patterns of diverse organisms. Furthermore, knowledge of population structure of genetic resources is necessary for the development of strategies for appropriate conservation of genetic diversity. Molecular phylogenetics and genetic diversity analysis can help to clarify the taxonomic identity and evolutionary relationships of the wild species. These methods can also help prevent misidentification and can carefully plan effective germplasm management strategies. Variability and genetic diversity are important factors in evolution and also in applied sciences because they determine the responses of a given organism to, for example, environmental stress, natural selection, and susceptibility to different diseases. Dear students, let's now discuss the importance of genetic diversity. Genetic diversity is a trait for both an individual or a population and is characterized by the percentage of heterozygous alleles in diploid organisms. Genetic blueprint is the fundamental of all living organisms that carry a specific genetic fingerprint. This is true irrespective of them being plants, animals, or fungi, whether they are short or long-lived, and whether they reproduce sexually or asexually. Therefore, conservation genetics and conservation genetics play a role to a large extent for the restoration of living organisms. Although the basic design underlying the conservation genetics may be very familiar, a very little attention has been made to genetic considerations with respect to conservation genetics. The genetic variability has an immense effect and has a great importance on the survival and reproduction of any organism as well as populations, which can be summarized as follows. 1. Effect on the individuals. Genetic variation or its deficiency within individuals can affect their survival and reproducibility. In diploid organisms, when both copies of gene are identical, that is homozygous at that gene or that locus or position, the expression of that gene may include traits having less beneficial for survival or reproduction in particular circumstances. This may cause several physiological or behavioral problems of genetic origin, such as malformed physical structure, poor biochemical balance, improper organ formation and function, altered social behavior, and susceptibility to different diseases. Homozygosity or lack of heterozygosity is the common result of inbreeding, that is sexual reproduction between closely related individuals or species, which leads to greater chance of two alleles of a locus being identical by descent from a common ancestor of both sides or by genetic drift. Dear students, small-sized populations that do not disperse or migrate well or are constrained allopatrically from gene flow with other populations can be particularly susceptible to inbreeding depression, which leads to reduced overall survival and reproduction of organisms with low heterozygosity. Inbreeding has a great impact on organisms' survivability and mortality, that is the death. It causes higher mortality, lower fecundity, reduced reproductive potentiality, reduced growth rate, developmental instability, and reduces adaptive ability to withstand environmental stress and lower competitive ability. Dear students, let's now discuss the effect on the populations. Genetic variability or diversity can influence any population in several ways. The growth rate of the population is lowered due to reduced fecundity and survival of the inbred individuals, which in turn increases the risk of extinction of the population concerned due to stochastic phenomena in near future. Lower capability for population growth also reduces the potentiality of a population to recover from population crisis due to several environmental stresses and genetic bottlenecks, which causes further inbreeding depression within the population. There are several factors that affect the population fitness. Therefore, if the population growth is not adequate to compensate the loss in fitness, a decline in genetic variation will eventually decrease the potential of the population to evolve in the changing environment. Dear students, Fisher in 1930s reported that the heritability of a trait is directly proportional to the evolutionary response to the selection, which in turn is proportional to the expected heterozygosity of gene diversity of that particular trait. Has reported that populations having genetic polymorphism in several alleles or having multiple alleles for a specific trait show great evolutionary adaptability. Thus, populations with lower heritability, reduced heterozygosity, and fewer polymorphic loci will preferably adapt more slowly and attain lesser adaptation than the population which is more diverse and having greater genetic diversity. Therefore, a sudden fluctuation in genetic variability in small populations can reduce the rate of adaptability sufficiently to cause small populations to go extinct in the face of environmental change to which large populations would likely be able to adapt. Every population should have an effective size to withstand genetic erosion and the loss of genetic variation due to genetic drift, genetic bottlenecks to preserve long-term survival potentiality. Mutations can basically influence the feasibility of small populations by bringing in inbreeding depression, by maintaining adaptive genetic variation in quantitative characters, and through the disintegration of fitness by the accumulation of mildly detrimental mutation. Lande in 1995 has suggested that highly detrimental mutations have a low effect to promote genetic variation because incorporation of new genetic variation is much slower than the overall mutation rate in populations. Therefore, considering the mutations are not detrimental, the effective population size needs to be in an order of 5,000 to ensure long-term viability of the population. So, random genetic drift is the major driving force that affects to change the allele frequencies in small populations. Therefore, deleterious mutations occasionally get fixed in small populations, replacing the more adaptable alleles. As the deleterious mutation accumulate in the populations, the population size may decrease and genetic drift becomes even more pronounced, commonly called as genetic meltdown. Dear students, let's discuss now that genetic diversity helps to adapt to environmental variability. Organisms live in complex environment that vary in spatial and temporal scale and is characterized by several factors such as weather, disturbance, events, resource availability, population sizes of competitors, etc. If a group of organisms were to live in a completely stable physical and biological environment, then a relatively narrow range of phenotypes might be optimally adapted to those conditions. Under these circumstances, organisms would benefit more by maintaining a narrow range of genotypes adapted to prevailing conditions, and allele frequencies might eventually attain equilibrium. By contrast, if the environment is patchy, heterogeneous, unpredictable over time, or includes a wide and changing variety of diseases, predators, and parasites, then subtle differences among individuals increases the probability that some individuals against others will survive to reproduce. That is, the trait of the organisms are exposed to natural selection. Since differences among individuals are determined at least partly by genotype, population genetic theory predicts that in variable environments, a broader range of genetic variation or higher heterozygosity will persist. Any population can tolerate the stochastic environmental variations through genetically controlled traits or characters. These traits are important from the standpoint of resistance and resilience ability of the population to tolerate freezing conditions, drought or inundation, high or low light availability, salinity, heavy metals, soil, nutrient deficiencies, extreme soil pH values, fluctuating temperature, dissolved oxygen, novel diseases in all groups of organisms. Plant populations often include individuals with a range of phenological calendar. For instance, Great Basin shrub populations include individuals that leaf out and flower over a period of weeks, increasing the likelihood of persistence of the population through periods of unusually early or late growing conditions. Knapp et al. In 2001 documented flowering periods in a population of individuals blue oak trees and found that trees initiated flowering over a period of a month in the spring. A diverse array of genotypes appears to be especially important in disease resistance. Genetically uniform populations are occasionally vulnerable to diseases and pathogens, and such uniformity also predisposes a population to transmit disease from one individual to another by direct contact or proximity. More diverse populations are more likely to include individuals resistant to specific diseases. Moreover, infected individuals occur at lower density, and thus diseases or pathogens may move more slowly through the population. Finally, genetic variation is a factor in competition among individuals in real ecological communities. Among animals, behavioral traits may regulate interspecific competition, since organisms make energetic or life history trade-offs among traits. For example, allocating energy between growth and reproduction, genetic variability is an important factor with regard to how populations function. Dear students, let's now discuss quantifying the genetic diversity. Genetic diversity is most often characterized using data that depict variation in either discrete allelic states or continuously distributed, that is, quantitative characters, which lead to different possible metrics of genetic diversity. The variation in allelic states or phenotypic traits may either be neutral or non-neutral with respect to fitness consequences. For example, molecular markers such as microsatellites, AFLPs, direct DNA sequences, or protein polymorphisms typically represent discrete allelic states that are assumed to be neutral. While molecular markers are probably the most common way that genetic diversity has been assayed in natural populations, neutral variation in and of itself cannot by definition have any ecological consequences. While neutral traits are typically measured as discrete allelic states, not all traits measured as discrete allelic states are neutral. In theory, quantitative traits may also be neutral, although in most cases researchers focus on quantitative traits with known or presumed functional significance. With discrete allelic states, metrics of genetic diversity reflect either the number of alleles or haplotypes within a population. For example, alleles per locus or summed allelic richness, and or the evenness of allele or haplotype frequencies. The latter can be measured by estimating the probability that two randomly chosen alleles or haplotypes in a population are expected to differ. So, given specific assumptions about the genetics and ecology of the population, that is expected heterozygosity, gene diversity, etc. A variety of other matrices can be also used to depict genetic diversity for discrete traits, and although they are often tailored to account for the type of inheritance of a particular genetic marker, for the most part they all reflect either the number, that is the richness, and or relative frequencies, that is the evenness of alleles. For quantitative traits, the measurement of genetic diversity begins by estimating the variance in a phenotypic trait among individuals that is due to genetically inherited differences. This involves either detailed knowledge about the pedigree of a natural population or a separate experiment that replicates multiple genetic families, that is SIB analysis, collected from a single population. The simplest measure of genetic diversity is total genetic variance in a trait, or VG, which is often further partitioned into its additive and non-additive components. Within a single generation, genetic variance for a given trait may have important ecological consequences regardless of its genetic architecture, in which case genetic variance is a relevant metric. Across generations, the key component for understanding how genetic diversity can influence the rate of evolution is the additive genetic variance. Additive genetic variance is used to estimate the narrow sense heritability for a trait which reflects evolvability, but heritability does not quantify genetic diversity per se because it is confounded by environmental variation. The coefficient of genetic variance is a more appropriate measure in this case. Dear students, let us now discuss the molecular tools to assess genetic diversity. The most powerful catalyst in the field of conservation has been the advances in genetic and molecular technologies, leading to a wide variety of molecular methodologies for application in conservation and population genetic studies. To date, molecular methods have been applied vastly in conservation biology, primarily as selectively neutral molecular tools for resolving the empirical questions of conservation and evolutionary relevance. The first step of molecular biology technologies in the field of conservation genetics was taken up in 1960s with the introduction of protein polymorphism analysis, followed by the mitochondrial DNA, that's mtDNA, restriction fragment length polymorphism, or RFLP based methodologies, randomly amplified polymorphic DNA, and more recently by the microsatellite marker based technologies, that is inter-simple sequence repeat, or ISSR markers, diversity array technology, and other high throughput platforms. Therefore, the application of particular types of genetic markers are becoming more and more specialized to achieve a particular goal to solve the specific questions of conservation concern. The genomic era was started when the genome of the evolutionary, ecologically, and commercially important model organisms were successfully sequenced. There are several benefits like larger number of molecular markers of a wide variety of organisms, SNPs, and microsatellite markers, high sophistication and additional use of neutral markers that enhance the fidelity of conservation study, enabling researchers to look deep into the problems regarding conservation concern. Expressed sequence tag, that is EST libraries, are the valuable resource for the study of conservation and evolutionary genetics using bioinformatic tools by Cascade databases called in silico or SNP mining pipeline More recently, genomic technological advances like next generation sequencing or NGS technologies and deep sequencing or ultra high throughput sequencing technologies provide a wide path to understand the empirical questions regarding the conservation genetics of model as well as non-model organisms. Dear students, let's now conclude the topic. The study of genetic diversity is very much essential from the standpoint of conservation and management purposes. Presently, Earth faces a huge threat to genetic diversity, especially to several endemic and threatened taxa. The genetic diversity of several species has gradually degraded and might be reaching a critical condition in near future. Several anthropogenic and environmental pressures are the main cause agents behind this phenomena. Therefore, the proper identification and estimation of genetic diversity of such critical threatened species is important for proper conservation and management purposes. Previously developed molecular tools and statistical procedures and recent advances in molecular genetics methods open a new door for better understanding the particular conservation and management unit and allow investigators to answer the empirical questions of genetic diversity regarding the conservation concern. The recent advances in computational biology, mathematical biology, biostatistics, and bioinformatics have opened a new window for the researchers to investigate the questions concerning genetic diversity and conservation in unequivocal way. Diversity clearly makes the world a more interesting place to live on, and the idea that we should conserve genetic diversity is therefore tempting. Yet, when we imagine imposing genetic diversity to secure the same good, its value is revealed as elusive, especially if we recognize it must be achieved at the cost of the well-being of some individuals whose existence has been used to produce a benefit enjoyed mainly by others. Dear students, it was all about today's lecture, Origin of Genetic Diversity. Hope you have understood it well. Take care. Goodbye.